Genome coverage bed.bg. No, you need a dash. Good. Okay, guys, really every single character has to be, you can't miss out these little dashes. You can't have anything else in here. It's, tab complete because it actually says it's there.
People might not actually know what tab complete is, so you can use demo it.
Sure.
Okay, but you're in the right place now.
So if I wanted to do this one function genome bed, so this, there's no, well, you can actually, but genome coverage bed, okay? And then I look at dash BG, dash IBAM, space, and then CTC, CT. If nothing comes up, then you know it's not there, CT. So then I could go tab, see, hit tab a few times, and it'll tell you what your options are. CTCF, and then underscore, PE, and then it says, okay, well, what's the next character? H, tab again. That's tab complete. So you put in the first few characters, you tab, and then it finishes it off for you. And if it's unique enough, it'll finish it off. If it's not, it'll say, here's what your options. And then so just a few more characters, tab it. Okay, yeah. Yeah. It's included in the program.
So which one are you loading here?
This is HG19. So can you guys see in the top corner here, make sure this says human HG19. That's the annotation that we used. And okay, so I loaded this data, the bigwig, everyone do that. Okay, and now we only have chromosome 22 in here. So just go into here and go to chromosome 22, and that'll zoom in to that chromosome. Okay, and then if you want to zoom in more, you can zoom in here. Okay. Zoom out. Okay, so you see there's a peak here, right? Yeah. From your Mac? Because what you're actually in a different computer. So you'd have to copy it onto your Mac and then load it. Yeah. But we're running everything within like some other computer. Yeah. Okay, did you load your bigwig?
So that's what we're asking. What's the file we load?
CTCF.bw. Sure.
You can do most things with most file types, and I think bigwigs. You can load bigwigs in CSC, right? Yeah.
It's meant for that because it's small and it's easy. You're good. Yeah. Right. You might just have to follow along for now. Okay. So what else can you do with this? So let's see. We're interested in. So also, if you want, I've provided this Synapse ID. If you want to go into your terminal and download it, you can do it that way. So if you're not in the X2Go client, just go synapse get ctcf.bw from your terminal, and it'll download the file for you. Okay, I've uploaded it to Synapse. If you want, if you didn't produce it yourself, you can do that. Also, if you didn't get the summits.bed, you can download that here. I've uploaded that. Okay. So in IGV, if you're interested in any gene, you can search in that region. So it's kind of like Google Maps, but for the genome. Okay? You can go to specific regions and look at it. So that you'd put that in here. So just, you see how it's human H19, you just put the gene name in there. So we're going to go to TBX1 because I know that that's on chromosome 22 and that's the only data we have. Okay? So just put it into this. Okay. So another thing that I added in here, I've loaded in that summits.bed file. That bed file that told me the coordinates of every single peak of where they should be. So here you could see this is the coordinate of that one peak that was called. It's right there. This is that other peak. So it's actually correlating. Oh, this one, yeah, it's correlating very well. Okay, so this is a great way to visualize your data. Now you could say, hey, ex-professor, I think that this transcription factor, CTCF, regulates TBX1 because there's a huge peak on the transcriptional start site, which is right here. And you could show them the data like that. You can't just say, here's chromosome, whatever, start and stop. It's nice to see visually, and like the coverage is how high this peak is. So you can see that you have a lot of peaks within this gene. So this is a great way to kind of visualize your data to present it to people. Okay? Okay, we're going to go to the next. And here's something that's important. You can actually also tell. You can also tell if. Oh, did I go to the beginning here? So here's two different antibodies that I used to pull down TBX5. Okay, the top one and then the middle one, and then here I've included the peak file for the top one. And this is also that narrow peak file that was produced by Max, included that kind of shows you the width as well. But anyway, here's two different antibodies. One's showing me nice peaks in different regions, and it's called nicely. The other antibody didn't show me a lot. It actually kind of just showed me garbage. So it's a good way to just kind of make sense of the data a little bit better as well. And don't forget, there's a ton of different kind of peak profiles. So depending on what you're really pulling down, if you're pulling down something like a chromosome modifier, you can kind of get a different profile. If you're doing transcription factors have usually a nice tight peak. So usually in the region of 50 base pairs. So you can get that peak profile. You can get really broad peak profiles. So it's important to kind of understand your data of like where like maybe this histone modifier is kind of regulating within the gene body. Whereas you can look at enhancers like there's H3K, I think 27, what is it? Not trimethylation, that's a repressive mark, but H3K4. You can look at markers that bind that are known to enhancers as well, so we're upstream. So you can actually say like, okay, it binds, this protein binds way upstream of this gene. Does it regulate it? Is it an enhancer? And then so you know the sequence of that. So if you wanted to even do a reporter of that enhancer, you know the sequence of where it is. So that this really is like, you know, it's interesting that there are such different types of peak profiles, and you really have to understand what you're looking at in order to assess it. Bed tools, also critical. This is really what kind of is a great kind of suite in order for you to manipulate your bed files. So if you wanted to say, I have these chromosome coordinates in this file, which ones are common? So you can run different tools to say, intersect these bed coordinates with this file's bed coordinates, and tell me the common ones. You can say which ones are distinct, which ones are like unique between them. There's also, you can also look at things like you can count the coverage. So if you want to know in this bed file, I want to know how many of these overlap. So it can kind of count how many reads are kind of within a region as well. So I'm just telling you, like, if you want to get a little bit better with manipulating bed tools and you had specific questions, like, look into this program because it's pretty important. Okay, so now that we know exactly where our peaks are, let's annotate them. Okay, so what? We have all these different transcription factor binding now. CTCF binds to all these different regions. What does that mean? Okay. So usually people would say, hey, if it's bound within the transcriptional start site, it probably regulates it because that's exactly where the transcription initiates, okay? So we're going to use Homer, which is another program from the SALC, and that's this. Let's get out of here. So we've installed Homer for you, and now I want you guys to type in this, okay? So this is it's calling the Homer program to annotate peaks.pl. Okay. So annotate peaks.pl, CTCF, and then you're going to do your summits.bed file, and then you're going to tell Homer, you're going to look at HG19, and then you're going to say, we're going to have we're going to input it as the bed graph. So if you made the bed graph already, and then we're going to hit return. Okay, so again, I'll put this up here. Please type this in exactly, annotate peaks.pl, and then you're going to put your bed file that you want to look at. That's the CTCF summits that we got from Max. Okay, that's the coordinates of all these peaks. We got to tell Homer that we're actually looking at the HG19 annotation. We're not looking at mouse. We're not looking at some other like newer or older annotation. We're looking at HG19. Then a dash, and this is all one, dash bed graph. Okay, all one dash bed graph, space, and then you put the CTCF bed graph file. So make sure, try to tab complete the file names, because then you know that you didn't type that incorrectly. Okay, how many people got it to run? Nobody got it to run yet? Oh, it's running. Shu, are you okay? Kenny, can you help Shu? Okay, so Homer completed, annotating the peaks. So let's look what it created. So we told them, take everything, the output of this whole kind of command, and put it as the CTCF homer.annotation, or underscore annotation. So let's just quickly head CTCF underscore homer, okay, tab complete, and here's a text file of all these different things. I'm going to try to load it up. Okay, so this is the output of the homer annotation. Okay, so you probably, let's see if I can, if that works. Okay. This is the homer anno.txt. CTCF underscore homer annotation. This is the one we just created with homer.
So to open this on theirs, they need to use, I think it is, Office and generic.
I could just kind of run through, the file. We're not going to go into this any further, just to show you kind of what the output is. So it tells you your peak, what the actual ID was. It tells you which chromosome. These are all going to be chromosome 22, obviously, because we didn't do anything else. The start and stop of the peak, what strand it's on, the score of the peak, so how big it was. The annotation, is it intergenic? Is it an intro? Is it a non-coding? Also, this is kind of interesting, the distance to the nearest TSS. So you think if it's within 2 kb of the TSS, that's more likely that these peaks are going to regulate it. And then, obviously, here's where you find your gene name. So this says, okay, so if you look, here's the gene IDs that were called for each of those different parts. So let's just quickly filter this data set, and let's look for something. Choose one between, let's say, greater than or equal to minus 2,000. So I'm just saying, okay, tell me all of these peaks that are between really close to the transcriptional start site. And then here's one that has a really high peak score, and it's pretty close. So we're going to go to this gene, and we're going to go to open up IGV, and let's look at it. Where's this peak? Okay. So it's right here. So likely, you can say, hey, CTCF probably regulates this gene because it's bound to the transcriptional start site. So, and then you can kind of go on from there. But that's a huge peak, because even if you look here, it's dwarfed by these other peaks that were called. Okay, so that's how you get the annotation. Yeah.
For the peak score, what's like the minimum group?
That's a good question, and because it does depend on your data too. I've looked at the top ones and then did the kind of looking at pathway analysis. But if I include more peaks, I get better results. And just because it's a small peak doesn't necessarily mean it doesn't do just as much as the bigger peaks. But sometimes, if it's a bigger peak, it could be more indicative of, or a broader peak, that it more highly regulates it. But yeah, it's kind of just look at the data and try to, it's like you're kind of, it comes with time just to see the data and say, hey, it's like everything. Okay. So we generated this annotation peaks, so we know, and we can actually take these lists and we can put them in. Do we want to do that? Let's just see. Copy. Let's go to top gene, which we learned yesterday. Let's go top fun, and let's see kind of maybe what does this regulate. Just hit return.
So you just pasted the top data?
Yeah, just the ones that were between 2 kb of the transcriptional start site. And, you know, you can kind of see maybe CTCF has to do with schizophrenia. Okay, maybe. And just kind of go through here and just try to see what's, you know, what's going on. Actually, schizophrenia comes up a lot. And then you could kind of look at what genes were called under that. Okay, so let's go back. And here was the output. Now, here's another program that I really like. It's called Great. And it's a browser-based program where you upload your peak file. And it does a lot of the grunt work and annotation for you. So it will go through and say, what's the nearest called gene, and what's it associated with. So really quickly, I'm going to do that. So great.stanford.edu. Okay, this is what the website looks like. I'm going to say you want the assembly again is HG19. It's this one here. It's human. Choose your bed file. So we're going to go to, let's see if I think I downloaded it. And also I put, I should have the Synapse ID for it as well. Let me see, it's not in desktop. I think it's downloads. You guys see anything that says great in it? Let's see. Oh, it's right here. Sorry, it's called great-summits. Here's the ID. Okay, so let's. I should have that somewhere. So if you can, download this. Can you see that? 5936606. So this is just the summit file with the coordinates. So it has the it'll say chromosome, which chromosome it's on, which is the start of the peak, which is the end of the peak.
What was the idea again? Sorry.
I have, let's see, where is that?
And all the commands are going into the Synapse page for this project. They should all be there now.
Yep. This is on Synapse. This is the ChipSEQ tutorial files. I think I still have to link it to the.
But it's on lecture 5, ChipSEQ 2. If you just click on that page on the wiki.
Quick question, how
Yeah.
did you even find that file, the chromosome 19 file?
So it's part of the Max testing files. So if you wanted to test out Max, they include the BAM files. So it's if you download the program, it's part of it. But usually if it's your own data, you do bow tie and then you get a BAM file. And then you work from there. The BAMs?
No, it's not going to the right direction. It's not going.
No,
to
you'd put BWs into there. So your terminal, you're on a different computer. Like you're doing your analysis over here, and then you're.
She's actually in.
She's in Next to Go.
That's Next to Go, yeah. This is Next to Go. It's just not going to MNT for some reason.
Oh, it's not?
IGB, yeah, it's a weird thing. We don't know why. Yeah. Yeah, if you do file, load file.
Okay, did everyone get this file? And then go to the browser. Go to your browser. Okay. Go to Great. Put your species, and then upload that bed file in there. Okay. That Great bed file. So this file is actually just your summits file, just the chromosomes start and stop. So it's just like a bed file that lists where your peak locations are. It doesn't include that extra, there's like an extra column or two that says the peak ID and the kind of score. So I just took out those two columns, and then you're only uploading the coordinates.
And sorry, is that something we generate, or do we have to download all the snaps?
Well, what you could do is you take your summits, put it into an Excel, and then just delete the two columns.
Okay.
And then, because actually, if you upload it to Great, it gives you an error saying, like, there's extra stuff in here. Yeah.
So if you uploaded the Synapse file to the X2Go client, how do you access that file?
So can you open up a browser within X2Go?
Ooh, that's a good question.
Yeah, so you go
It has
to,
Firefox installed.
you go to there and you go to internet. What was the gene you looked at?
TBX1.
TBX1. So here you type in TBX1. And there's speaks. And you can zoom in and out.
Okay, did everybody upload that file? Okay, and then do the genome background, the next one. So I'll go back. Whole genome as the background, and then hit submit. Tell me what you get. Is it working? Okay. So again, this is helping you understand your data. You're annotating it. What are the kind of major Go pathways that are coming out of it? Okay.
So in your, the Gray Summit file,
Yeah.
can you just kind of quickly cap, where did you find that Gray Summit?
So that's actually it comes after you run Max, you get a chromosome it tells you where all your peaks are. So it says your chromosome, your start and your stop. So that's what that is. It's just those three columns. So you can open up that file in the summits.bed from Max, delete the last two columns, and then you get this Great Summits. So, because Great actually has a problem if you have extra kind of columns in it. So you just want the chromosome, the start base pair, end base pair, and then upload that file, a column, 3 columns. Okay. So if you look again here, let's see if we get any schizophrenia coming out here. Anybody see any? Oh, right there. So disease ontology. So this is another way, but there's a ton of other kind of things in here. Panther, MSIG-DB. It's just like a one-stop shopping kind of thing for ChipSEQ if you want to annotate it. Okay. Perturbation. Here's some kind of PubMed things that have talked about it. the p-value. Okay? So you can, yes?
Are there any programs that allow you to map motifs to identify the
Yeah.
strengths and
Yeah. So that's right next. If we can, if we have time, do we have time?
3 minutes.
Okay. So since we only have 3 minutes, I'm not going to talk about making like kind of more publication figures for it. Let's go straight into motif analysis. So there's a few ways you can do this, and I'll just talk about this way, this one way that I like a lot. But you can use bed tools. And what you could do is now that you know exactly where the peaks are, where the exact kind of base pair of within the genome of where it is. So I use bed tools to kind of say go 50 base pairs up and 50 base pairs downstream. So if you're looking at transcription factors, you kind of want like 100 base pair window in your chromosome coordinates. So bed tools can do that. That's what this does. And then get FASTA, that's really important. So get FASTA takes those chromosome start and stops of those 100 base pairs, and it looks at an annotation of those regions and pulls down the sequence. So it'll say from these coordinates, and let me quickly download that file because I think it's interesting to look at. FASTA.out, it's this one. So again, so this step was just, it's important for taking your coordinates and getting the sequence of that region. Okay. So here you're seeing I gave it a bed file that contains chromosome 22 and then about approximately 100 base pairs within that region. So that, and then it pulled down the sequence in that region. So then you can plug this into programs to say algorithmically, is there a motif in here? And you can go to, there's another browser-based version of this. You can input into there, and you can get these results. So no time to go through that. But it's called MemeChip. Okay, so, and also let's use Homer because we're kind of a little bit more familiar with Homer right now. And let's type this command in. Okay. So paste. So we're going to go find motifs, genome, and make sure, again, tab complete, and you got to capitalize the M, the G, .pl, space, ctcf summits.bed, hg19, space, and then you put the output directory. So let's say, maybe it'll work right within this. No. So make, what's that? Dot. Okay. So it's running. So it's saying, you know, it's going to go through, oh, I should have done a few more things because this is going to take a while. So this is the default values to find the motifs. So it's actually, you're going to say find the top 25 motifs that we think it is. You know, and where are they located? What's de novo motifs? And then it's actually going to compare it to the known database of the motifs and then give you a p-value of that, and then you could kind of comb through that. This does take a long time. And you can actually say, hey, I want to look at motifs that are 8 base pairs, 9 base pairs, 10 base pairs, you know. So there's all these different options within Homer that you can use to find the motifs. So I'm just going to stop this but at least maybe when we when I upload the presentation, you guys will see all the different command lines so you can do it at home. And then let's just go in and look at that kind of results directory. So if you guys want to still continue, just to run it for yourself. But again, it took that bed file from max. So everything starts out with max, and then you get these kind of coordinates of all your peaks, and then you can go from there. Okay, so let's see. Box downloads. So, and here's the results. So after it's done, and then you can go to this known results. So this matches your motifs that it called to known motifs in the literature. And let's open that up, see if it works. Okay. So you'll see the top ranked motif is CTCF. And I've run it through a couple other kind of things, and it gives you the motif. So if you're ever going to present this, you'd show this kind of this logo here and saying that's the motif for CTCF. And it called it, and here's the p-value. So therefore, this is likely CTCF. But you can, if you don't know kind of what the transcription factor is that binds in these regions, you can kind of go through the other ones and also look, because it'll call other ones. And if it's and it makes sense if it's a longer kind of region also that it has a motif in, there could be multiple different factors that all come together to bind to that region. So you can look through these different kind of motifs that are known and try to make sense of your data to say what are the transcription factors kind of regulating these regions. Okay, so I did. So for Homer, I did not, but you can. But for I was going to show you that meme chip. You take your chromosome coordinates, and then you kind of take the start, and then you do 50 base pairs up, so minus 50, and then your end, and you do 50 base pairs forward. So you have now your input is chromosomes, and the start and stop are 50 between, or 100 between. So you can define exactly how much regions you want to look at. But if you just wanted to do a default kind of thing, like a lot of these programs are very smart, and they'll kind of give you some good results fast. So if you wanted to throw together a quick poster, just copy paste, p-value, great, publish. Okay, does anybody have any other questions on it? Because that's anything else? Stylistic. Some of them are rapidly developed over time. Some of them kind of drop out. I think meme was better initially. Homer's getting a lot better with every iteration. I actually use both of them, and then I try to see what overlaps. So stylistic, whatever you want, whatever gives you what you're looking for. Okay, that's it. So let me know if you guys had any further questions offline, and be happy to answer any questions.
Could you provide slides that have those addresses for those?
It should be linked to the.
And I already